So, how are you all? Welcome back to the JP classes. Myself, Julie Sharma, is back again with the chapter 2 of Biology of CBSE Board. The chapter is Sexual Reproduction in the Flowering Plants. So, let's start. You can follow us on Facebook, Instagram, and you are also seeing some contact numbers and mail ID. You can also contact us here. So, now moving to the topic. First of all, the chapter itself is Sexual Reproduction in Flowering Plants. So, first of all, what are flowering plants? Angiosperms are considered as flowering plants because they have the special characteristics of bearing flowers. Now, what is flower? A flower is a modified shoot which develops on the plant system, or you can say a plant body. A flower is also a sexual reproductive part of the angiosperms which develops after the vegetative phase. Now, a flower is the one which later on give rise to the seeds and the fruits. We will see it later, but now before starting we should have a brief description of a flower. So, they are the main four walls, or we can say that four floral members of a flower which are arranged on a central axis. The central axis is this one, which is known as the thalamus or the receptacle. This is the basal this part on which the four walls are attached. These are known as the thalamus or receptacle. Now, starting from the outermost one, the first and the outermost accessory wall which comprises of the sepals. So, you can see here it is written the sepals. These sepals together known as the calyx, which forms the outermost first accessory walls of a flower. What is the role of a sepal? The sepals, or we can say the calyx, is the outer one which protects the flower in the bud stage. Besides that, if they are green, you find that when you open a flower, you find the small green leaf like structures outside the flower. These are the sepals. When the bud is opening, these flowers get open and gives rise to the flowered open inflorescence. So, the role of a sepal is to provide the protection to a flower at the bud stage, and besides that, the other function is when it is green, it performs photosynthesis, and if it is non-green, or we can say colored, it provides the attraction to the insects by giving the fragrance, odor, smells, etc. Now, moving to the inner wall, that is your consisting of the petals. So, the next wall which is inner to the petals is inner to the sepal is your petals. The wall comprising of a group of petals is known as corolla. So, now what is the role of corolla? Actually, these corollas are greater in size, or you can say bigger in size as comparison to these sepals. Petals are generally brightly colored, they are scented, they are fragrant, so as they can attract the insects for the pollinations, or we can say the reason for the bright color is to attract the insects which act as a pollinators. I think you know about the pollination and the pollinators terms because I have discussed in the previous lecture. Now, inner to the petals, some flowers bears the different roles of petals or corolla. Besides having or attracting the insects as the pollinators, they also have some glands which secretes the nectar. You all know that insects feed on the nectar, and they mainly the honeybees, if you are asking, the honeybees which feed on the nectar and gives a very highly nutritive product known as the honey. So, it is because the petals are providing you the glands which secretes the nectar. Now, moving to the petals inner, you find that stamens. a wall comprises of the group of stamens is known as androecium. Androecium is considered as the male reproductive part of a flower, which comprises of two parts. You can see these are your stamens. This is your individual stamen. Okay, this bilobed structure. The focus on the term bilobed. Bilobed is because it is having the two lobes. One lobe is this one and second lobe is this one. The bilobed structure is known as the anther, and the anther comprises of the pollen sacs where your microspores known as pollen grains are residing. So, what is the role of a pollen grain? The pollen grains are the structures which are comprising of the male gametes which fertilize with the female gametes to forms the zygote. Now, moving to the next part, the anther is attached through the filament. So, what is a filament? It is a stalk-like structure which is connecting this anther portion to the receptacle via a stalk known as your filament. I think it is clear now. So, you can say that stamens are the male reproductive parts which are giving you the pollen grains, and pollen grains are comprising of the two male gametes. Now, moving to the innermost wall, or we can say that the female reproductive part of a flower, which comprises of a group of pistils which are also known as the carpels. पिस्टल्स का अदर नेम कार्पल्स भी होता है, और ये कार्पल्स जो होते हैं ये 3 पार्ट्स फॉलो करते हैं. द अपर मोस्ट पार्ट, दिस पार्ट विच इज़ हैविंग you can say a cushion like structure which help in collecting or taking up all the pollen grains which have come either from the pollinators, either from the wind, either from the water or anything. They resides on this stigma. Now, the stigma is followed by this stalk-like structure. This stalk-like structure, it is known as your style. So, what is style? A style is a stalk which is connecting this stigma to the basal part which is known as ovary. So, stigma and ovary are connected through a stalk, this stalk. I am talking about this stalk, right? This stalk is known as the style. Now, I think these parts are clear to you. Moving on to the basal swollen part or the innermost part of the pistil. What is this innermost part of the pistil is? This is your ovary. You can see this is a basal swollen portion. You are seeing like this cup-like shaped structures. These are your ovary, and inside the ovary you can see these small round structures. You can see now these are known as the ovules. These are known as the ovules. These are the different parts of a flower. Now moving to the next slide. Now moving to the next slide, that is a flower. I am going to tell you few important points of a flower. So first of all, what is a flower? It is a modified shoot. Now second thing, now moving to the next slide, that is flower. I am going to give you a few short characteristics of a flower. So the first character is a flower is known as a modified shoot. What is shoot? Shoot is that part of the plant which is above the soil level. Okay. Now second point is it develops from the floral primordia. What is floral primordia? Floral means the part which is giving rise to a flower, and primordia is a small outgrowth or a small basal part which indicates that from this portion the flower will appear. Now next, what is the primordia will develop into the floral bud and then into a flower. We all know that before the flower arises, a bud stage appears. Now next, it is a reproductive part of a plant, androecium. I have already told you, androecium is a male reproductive wall which consists of the stamens, and gynoecium is a female reproductive part which comprises of the carpels. So, is it clear? Now, the next is what are stamens. Andrecium comprises of a group of stamens, and stamen is the basic unit which comprises of two parts. So we can say that stamen is a male reproductive unit, okay, because it comprises of the pollen grains which comprises of the two male gametes. Now the stamen consists of the two parts. One is your filament, the stalk one. This is this stalk is known as your filament, and the anther. Anther what? Anther, this is your bilobed structure. This structure is known as your anther. Now filament is attached to the thalamus or the petal. These filaments help this anther to attach to either to the thalamus or the petals, because in some flowers we see that the petals are fused with the andrecium part. So in that case it is attached to a petal. Now moving to this one, this is the basic structure, or you can say the transverse section of the anther. I have already told you anther is a bilobed structure. One lobe is this one and second lobe is this one. These two lobes are separated by a group of tissues known as your connective tissues. Means how can you differentiate that this is the first lobe and this is the second lobe? To differentiate between these, a group of cells are, or you can say a group of tissues known as the connective tissues is present, which demarcates the two lobes. Now in each lobe the two pollen sacs are present. Pollen sac are the compartments which comprises of the pollen grains. So and this is your line of dehiscence. What is line of dehiscence? The line at which they when they get mature, the humidity will decrease, leading to a pressure on the wall which gives rise to the opening of the pollen sac and releasing the pollen grains into the environment. So it is known as the line of dehiscence. So you can see that these two lobes are comprising of pollen sac. Each lobe is containing two pollen sacs. So we can say anther is comprising of totally 4 pollen sacs, and pollen sacs are comprising of thousands or hundreds of pollen grains which consist 2 male gametes. Now moving to the next slide. Now I am showing you the diagram explaining the transverse section of the pollen sac. Now this is your anther, ठीक है, ये anther के आपके ये बीच में ये जो है, this is the separation of the two lobes, this is the one lobe, this is the second lobe, and this you can find out, this is your connective tissues. I told you na, the two lobes are separated by the tissue known as the connective tissue. This is the one. Now each anther is surrounded by mainly 4 layers, ठीक है. Starting from the outer one which is a single layered cell comprises of the epidermis. जो बाहर वाली आपको single layer दिखती है उसी को हम epidermis बोलते हैं, which is protective in nature, वो protection का काम करती है. उसके अंदर जो आप देख रहे हैं उस वाली लेयर को हम बोलते हैं एंडोथीशियम. ये भी सिंगल लेयर्ड होती है. इट इज़ ऑल्सो प्रोटेक्टिव इन नेचर. समटाइम्स इट इज़ ऑल्सो सर्विंग एज अ स्टोरेज पर्पज़. ये स्टोरेज पर्पज़ का काम करती है. Inner to the one lies the multiple number 2 to 3 layers of cells known as your middle layers, and the inner one which directly linked to the pollen sac is the tapetum. It is a most important question in the board exam. Name the innermost layer of the pollen sac, or you can say that what is the function of the tapetum. The tapetum is a cell which provides the nourishment to the developing pollen grains which are present within the pollen sac. Now this is clear. After this you can see these 4. Now after this I want to bring your attention to the most important thing. What is Microsporangia? Actually you can see here in this diagram. You can see these 4 oval or somewhat spherical shaped structure. These are earlier known as the. I want to bring your attention what is Microsporangia. Actually you can see in this diagram, that is anther one. In anther you can see these 4, right? You can see these na? these 4 spherical structures are earlier known as Microsporangia. You can say that one anther comprises of the 4 Microsporangia, which later on develops into the pollen sac. Now I am taking this single pollen sac and explaining you the whole structure. The pollen sac is comprises of 4 layers. The outer one is known as the epidermis. You can see the epidermis is this layer. This single layer is known as epidermis. Inner to the one lies a single layer of cells which are closely compact. These are known as your endothecium. Inner to one lies a 2 to 3 layered cells known as your middle layers, and the inner one, which is in direct contact with the pollen grains, is known as the tapetum. This is your tapetum. It is this lining is your tapetum. You can see that the tapetum is having the dot structure. These are nothing but the chlorophylls which are able to perform photosynthesis, as well as they are nutritive in function. Now besides that, you can see some cells are showing the nucleus. These are the microspore mother cells. What do you mean by microspore mother cells? In short way, you can see MMC. Microspore mother cells are the ones which are going to give rise to the microspores. Means microspore mother cells वो parent cell है जो meiosis करेगा और क्या बनाएगा, आपके microspores बनाएगा. Now moving to the next slide. This is the diagrammatic or colorful sketch of the earlier one, the anther. This is the anther. This particular point is showing that it is from here the filament is attached to the anther. These are your 4 pollen sacs which are comprising of the these dot structures known as the pollen grains, and you can see that after this, when the pollen grains get matured, the humidity level, or you can say the moisture level, becomes less, leading to the pressure on the walls, causing the dehiscence of the anther, and the pollen grains are released in this way. Now moving to the next slide. Now I am giving you some points about the Microsporangia and the anther. First point is, you know that the structure of anther, that is known as the Microsporangium, because the anther comprises of the 4 Microsporangium. So anther is a bilobed and dithecous. What is theca? Theca means covering. They are having the 2 coverings known as the theca. Now a longitudinal groove separate the theca, means बीच में एक lining होती है जो 2 आपके thecas को divide करती है. That is known as the separating the theca. In a cross section, anther is a tetragonal structure. Tetragonal means which are having the 4 sides. You can see ना anther 2 ऊपर pollen sac थे, नीचे 2 थे, जिसकी वजह से ऐसी shape आ रही थी. इस shape को हम tetragonal shape बोलते हैं. Consisting of the 4 microsporangia, 2 in each lobes. Later, these 2 microsporangia of each lobe fuse as a pollen sac. These 2 pollen grains के जो 2 microsporangia थे, each lobe में वो fuse करके pollen sac में convert हो जाते हैं. ठीक है? Now each microsporangium is circular. I have shown you in the previous slide the diagram of this one. A microsporangium is circular, surrounded by the 4 layers. These layers are starting from the outer one, epidermis, endothecium, middle layers and tapetum. The outermost layers protect the pollen and help in the dehiscence of anther to release the pollens. मतलब जो आपकी बाहर वाली layers होती हैं, जो protective in function होती हैं, जो जब ह्यूमिडिटी कम होती है तो वो डेहिसाइज करती है, pollen grains को रिलीज करती है. नाउ मूविंग टू द नेक्स्ट. नाउ द इनर मोस्ट वन, जिसको हम क्या बोलते हैं, टेप्टम, इट नरिशेस द डेवलपिंग pollen grains. What do you mean by the developing pollen grains? The pollen grains which are generating, which are producing within the pollen sac. These are known as the developing pollen grains. So the tapetum is providing the nourishment to them. When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangia. कहने का मतलब है, सपोज करो एक microsporangium है उसके सेंटर की कुछ सेल्स जो होती हैं वो स्पोरोजेनस टिश्यूज़ फॉर्म करती है, क्योंकि बहुत सारा रिजर्व फूड उसमें अटैच रहता है, और उन्हीं में से कुछ सेल्स जो होती है वो माइक्रोस्पोर मदर सेल्स की तरह बिहेव करना शुरू कर जाती हैं। तो ठीक है, नेक्स्ट। Now, you have now studied what is anther, what is the pollen sac, what is microsporangia, and after that you are listening continuously pollen grains, pollen grains, pollen grains. Now I am going to explain the structure of the pollen grains. This is a structure of a pollen grain. This is a two-celled structure. It is known as a two-celled structure because it is comprising of two cells. This one is a one cell which is larger in size, and this is a second cell which is smaller in size. The bigger cell, which is this one, it is known as your vegetative cell, and the lower one, this one, it is known as your generative cell. Means we can say that जो north direction की तरफ है, जो बड़ी size में cell है, जो वेजिटल पोल की तरफ है, उसे हम क्या बोलते हैं, आपकी वेजिटेटिव सेल, और जो नीचे है, जो छोटी सी आपकी स्पिंडल शेप्ड ये जो दिख रही है, इसको हम बोलते हैं जेनरेटिव सेल। द जेनरेटिव सेल जो स्मॉल साइज की है, इट इज़ द मेन सेल विच इज़ गोइंग टू गिव राइज टू द टू मेल गैमेट्स। अब उसके अलावा जो सेल है आपने देखा वो दो लेयर्स, दो आउटर कवरिंग से कवर्ड है। जो दो आउटर कवरिंग्स हैं, द बाहर वाली जो आप देख रहे हैं जो इर्रेगुलर सरफेसेस से रिप्रेजेंट है, कहीं मोटी है कहीं पतली है, इस बाहर वाली लेयर को हम एक्साइन बोलते हैं। ठीक है, जो एक्साइन है वो इर्रेगुलर होती है, कहीं से वो मोटी होती है, कहीं से वो पतली होती है, इसीलिए उसकी रफ स्ट्रक्चर होती है। ठीक है, बिसाइड्स दैट जो और ये जो आपकी एक्साइन होती है, इसकी जो इम्पॉर्टेंट कैरेक्टरिस्टिक है ये कि ये एक केमिकल से कॉन्स्टिट्यूएंट, इसका जो कॉन्स्टिट्यूशन है वो एक है केमिकल जिसका नाम होता है स्पोरोपोलिनिन। स्पोरोपोलिनिन क्या है, वो एक केमिकल है जो रेजिस्टेंट है केमिकल्स के लिए, जैसे ऑर्गेनिक एसिड्स हो गए, आपके एसिड्स हो गई, एल्कलीज हो गई, हाई टेम्परेचर, इन सब से रेजिस्टेंट है। मतलब इन सब का इस पर कोई इफेक्ट नहीं है, जिसकी वजह से आप पोलन ग्रेन्स को एक काफ़ी लॉन्ग टाइम की तरफ प्रिजर्व करके रह सकते हो। दैट्स वाई दे आर ऑल्सो कंसिडर्ड एज अ फॉसिल्स। आप सबको पता है फॉसिल्स क्या होते हैं? फॉसिल्स वो होते हैं जिनको हम सालों करोड़ों सालों तक ऐसे कैसे मेंटेन प्रिजर्व कर सकते हैं। वैसे ही ये जो पॉलेंस हैं इनको हम प्रिजर्व कर सकते हैं बिकॉज जो इनकी एक्साइन लेयर है वो किससे बनी होती है? एक हार्ड मटेरियल से, एक रेजिस्टेंट मटेरियल से, जिसका नाम है स्पोरोपोलिनिन। ठीक है, ये क्लियर हो गया। नाउ मूविंग टू द जो एक्साइन के अंदर जो आप लेयर देख रहे हो, जो एक स्मूथ लेयर है जिसके डायमीटर यूनिफॉर्म है बिल्कुल, इस लेयर को हम बोलते हैं इंटाइन। ठीक है, जो सेलुलोज और पैक्टिन्स की बनी होती है विच इज़ इजीली डिग्रेडेबल। एंड जो ईच सेल है उसमें आपने देख रहा है कि दोनों में एक एक सिंगल न्यूक्लियाई प्रेजेंट है, साइटोप्लाज्म प्रेजेंट है और उनकी ये सेल मेम्ब्रेन है। तो दिस स्ट्रक्चर इज नोन एज द टू सेल्ड पोलन ग्रेन। नाउ देयर इज़ ऑल्सो सम मोस्ट ऑफ दी जो एंजियोस्पर्म्स हैं जो अपने पोलन ग्रेन्स को शेड करते हैं वो टू सेल्ड स्टेज पे करते हैं, लेकिन कुछ प्लांट्स ऐसे हैं जो पोलन ग्रेन्स को थ्री सेल स्ट्रक्चर में शेड करते हैं। तो थ्री सेल बनेंगे कैसे, मैं आपको वो समझाने जा रही हूँ। ये जो जनरेटिव सेल है, आप सबको पता है जब भी डिवीजन होता है तो वो दो स्टेप्स में होता है, एक होता है कैरियोकाइनेसिस, एक होता है साइटोकाइनेसिस। सबसे पहले क्या करेगा, ये जो आपका जनरेटिव सेल का न्यूक्लियाई है वो डिवाइड करेगा। डिवाइड जो होगा वो माइटोटिक डिवीजन होगा, क्योंकि हमें क्या करना है, प्लाइडी को सेम रखना है। अब जब ये न्यूक्लियस डिवाइड करेगा तो दो न्यूक्लियाई बन गए। ऐसे ही जब न्यूक्लियर डिवाइड हो जाएंगे फिर जो ये साइटोप्लाज्म है वो भी डिवाइड होना शुरू हो जाएगा, उसके बीच में एक वॉल बन जाएगी जिसकी वजह से यहाँ पे क्या होगा, यहाँ पे एक सपोज करो एक सेंटर में वॉल बन गई, ये दो आपके अलग अलग सेल्स बन गए, तो टोटल कितनी सेल हो गई, एक, यहाँ पे दो और यहाँ तीन। इसीलिए इस स्ट्रक्चर को हम थ्री सेल्ड पोलन ग्रेन स्ट्रक्चर बोलते हैं। इतना क्लियर हो गया। नाउ आपने देखा होगा जब भी कि हम कहते हैं कि पोलन ग्रेन्स स्टिग्मा पे पहुँच चुका है, अब वहाँ पे क्या होगा, वो जर्मिनेट करेगा। तो जर्मिनेशन का मतलब क्या है? जर्मिनेशन का मतलब होता है कि अब वो पोलन ट्यूब बनाएगा। अब पोलन ट्यूब क्या है? पोलन ट्यूब एक वो ट्यूब है या वो आउटग्रोथ है जो जर्म पोर से अराइज होगी। तो अब हमें पहले हम ये पढ़ें कि पॉलन ट्यूब क्या है, उससे पहले हमें ये पता होना चाहिए कि जर्म पोर होता क्या है। तो जर्म पोर एक्चुअली है क्या? जब आपने देखा कि जो एग्जाइन है यहाँ बहुत पतली है, इतनी पतली है कि ये फ्यूज़ हो रही है इंटाइन के साथ, तो ये जो पर्टिकुलर पॉइंट्स हैं जहाँ पे एग्जाइन फ्यूज़ होती है इंटाइन के साथ, वहाँ पे एक छोटा सा पोर बनता है जिसको हम जर्म पोर बोलते हैं। तो इसी जर्म पोर से जब पॉलन ग्रेन्स जर्मिनेट करते हैं तो पॉलन ट्यूब देते हैं, और उस पॉलन ट्यूब में क्या ट्रेवल करते हैं? आपके 2 मेल गैमीट्स जो ओव्यूल्स को रीच करने के बाद एग से फ्यूज़ करते हैं। आई थिंक इज इट क्लियर टू यू? नाउ मूविंग टू द नेक्स्ट स्लाइड। नाउ after that I would like to explain you the term microsporogenesis. The word can be divided into 2 parts. This one is known as, this is micro and this is genesis. Microsporogenesis comprises of the 2 parts. Microsporo means microspores and genesis means formation. Microsporogenesis वो process है जिसमें हम microspores की formation करते हैं. And what are microspores? Microspores means pollen grains. So now the process of formation of microspores from a pollen mother cell, जिसमें मैंने आपको बताया था उसे microspore mother cell भी बोलते हैं, जिसे हम MMC shortcut में यूज़ करते हैं, through the meiosis is called the microsporogenesis. The cells of the sporogenous tissue undergo meiosis to form the microspore tetrad. What is tetrad? आप सबको पता है जो meiosis है वो 2 parts में divided है, meiosis first and meiosis second. तो total जो result होता है meiosis का वो 4 haploid cells बनते हैं, और वो एक linear way में एक के बाद, जैसे ये है मेरा हाथ है, 1 सेल 2 सेल 3 सेल 4 सेल, तो ये चारों लीनियर फॉर्म में होते हैं, इसे हम टेट्राड बोलते हैं। तो क्या होगा, जो आपके स्पोरोजेनस टिश्यूज़ हैं वो क्या करेंगी? मियोसिस करेंगी और 4 आपके सेल्स फॉर्म करेंगी जिसे हम माइक्रोस्पोर टेट्राड बोलते हैं, विच आर अरेंज्ड इन अ क्लस्टर ऑफ 4 सेल्स। नाउ एज ईच सेल of the sporogenous tissue has a potential to form tetrad. Sporogenous tissue की each and every cell के पास वो capability है कि वो एक pollen mother cell की तरह act कर सकती हैं. That's why they are also known as the microspore mother cell. On maturation and dehydration. Maturation तो आपको पता है कि वो mature होगा. Dehydration का मतलब मैंने भी बताया था, जैसे हम लोग ही हैं, हमारे अंदर एजिंग होती है तो आपने देखा कि हमारी जो स्किन है वो सिकुड़ने लग जाती है, वो लूजन अप होने लग जाती है, जो उसकी वो टाइटनिंग होती है वो लूज़ हो जाती है। सिमिलर वे में क्या होता है, जब पॉलन ग्रेन्स मैच्योर होते हैं तो जो मॉइस्चर कंटेंट है वो रिड्यूस होता है। मॉइस्चर कंटेंट रिड्यूस होगा तो सेल्स अपार्टली लूज होंगे, दूर दूर भागेंगे, जिसकी वजह से एक गैप क्रिएट होता है जिसे हम डेहिसेंस बोलते हैं। ठीक है, तो on maturation and dehydration of anther, the spores dissociate and develop into the pollen grains. Pollen grains release with the dehiscence of the anther. कहने का मतलब है pollen grains कब रिलीज होंगे? जब आपका dehiscence होगा किसका? एंथर का। मतलब जब आपके एंथर्स में आपके गैप्स क्रिएट होंगे, उन गैप्स के थ्रू आपके pollen grains एनवायरनमेंट में रिलीज होते हैं। Now moving to the next slide. Now pollen grains जिसे हम मेल गैमेटोफाइट भी बोलते हैं, मेल बिकॉज इट इज रिप्रेजेंटिंग द मेल पेरेंट, एंड गैमेटोफाइट इज द पार्ट विच इज गोइंग टू गिव राइज टू द पर्टिकुलर गैमेटोफिटिक स्टेज, इसीलिए इसको हम pollen grains को मेल गैमेटोफाइट्स भी कंसिडर करते हैं। नाउ इनकी अगर हम स्ट्रक्चर्स की बात करें, दे आर स्फेरिकल इन शेप। नाउ they have 2 layered wall, outer hard exine layer and the inner is thin intine layer. Exine, it is made up of sporopollen. Please focus on this term, it is very important from the board exams. Sporopollen, it is resistant to the organic matter, it can withstand the high temperature, acids, alkalis and enzymes. It has a prominent apertures. Apertures means spaces known as germ pores. This term is germ pores where the sporopollen is absent. Sometimes you can also define the germ pore as these are those positions within the pollen grains where the exine is absent, or you can say where the sporopollen is absent, क्योंकि आपको पता है exine sporopollen की बनी है, अगर exine नहीं है इसका मतलब sporopollen भी वहाँ पे present नहीं होगा। Now the intine, which is of thin continuous layer made up of cellulose and पेक्टिन्स। ये सारे पॉइंट्स हम ऑलरेडी डिस्कस कर चुके हैं, लेकिन डिटेल में आप लोगों को बताने के लिए मैंने यहाँ पे मेंशन किए हुए हैं। नेक्स्ट, मूविंग टू द नेक्स्ट स्लाइड। नाउ pollen grains contains the cytoplasm which is surrounded by the plasma membrane. Mature pollen grains has two cells, that is the vegetative cell and the second one is known as your generative cell. मैंने बताया था जो वेजिटल पोल की तरफ होती है, जो ऊपर नॉर्थ पोल की तरफ होती है, जो बड़ी होती है साइज में, उसे हम क्या बोलते हैं, वेजिटेटिव सेल, और जो नीचे लोअर टुवर्ड्स द साउथ पोल होती है, small in size होती है, which has the function to form the two male gametes, it is known as your generative cell. The vegetative cell is bigger, abundant of food reserve, large irregular nucleus, whereas the generative cell is small, spindle shaped. Why it is spindle shaped? आपने देखा सेंटर से उसकी जो डायमीटर थी वो ज़्यादा थी, और जो एंड्स थे वहाँ पे उसकी डायमीटर क्या हो गई थी, डिक्रीज हो गई थी, जिसकी वजह से उसने एक स्पिंडल शेप ले लिया था. With the dense cytoplasm and the nucleus floats in the vegetative cell cytoplasm. In 60% species, pollen grains are shed in the two-cell stage. Two-cell stage का मतलब एक आपकी एक आपकी vegetative cell, एक आपकी generative cell. इस stage को हम two-cell stage बोलते हैं. Whereas the 40% species shed in the three-cell stage. How the three-cell stage is formed? In which the generative cell divides mitotically into the two male gametes. मैंने आपको पहले भी बताया था जो डिवीजन होगा वो माइटोटिक होगा. क्यों? क्योंकि हमें प्लॉइडी को क्या रखना है, सेम रखना है. तो अब जब माइटोसिस होगा तो दो डॉटर सेल्स होंगे, तो दो तो ये हो गए और एक आपका वेजिटेटिव, तो टोटल कितने हो गए, 3, और इसी स्टेज को हम 3-cell stage बोलते हैं. नाउ आई वुड लाइक टू स्टॉप हियर ओनली. This is the overall topics that we have discussed. The rest topics will be discussed in our next lecture. Till then, take care. See you. Bye bye.